In the present times, wherein the coronavirus with just 30,000 base pairs have brought the lives of humans with 30 million base pairs to halt, nothing more is needed to emphasize the importance of discovering viruses that can hinder human lives. One such momentous discovery was that of Hepatitis C virus, for which the Nobel Prize in Physiology and Medicine 2020 was awarded to. The award was shared by three diligent virologists, Harvey J. Houghton and Charles M.Rice, who discovered, identified, and understood the potency of Hepatitis C virus. Now, let's have a look through the milestones which led to this life-saving discovery. So, what is hepatitis? Before understanding what hepatitis C is and how it was discovered, let's first comprehend hepatitis and the viruses causing it. Hepatitis is inflammation of the liver that happens when tissues of the body are injured or infected, which can damage your liver. Hepatitis can be caused due to toxins, autoimmune reactions, viruses, and several other causes. The main cause of viral hepatitis are hepatitis A, B, and C viruses. Hepatitis A is an acute disease, while B and C are often chronic viruses and disease which may progress into cirrhosis or even liver cancer. Discovery of hepatitis A and B viruses. Hepatitis A virus, discovered in 1973, is transmitted through ingestion of contaminated food, water, or through direct contact with an infectious person. Well, a safe and effective vaccine is available to prevent hepatitis A. Hepatitis B is a viral infection that attacks the liver and can cause both acute and chronic disease. The virus is most commonly transmitted from mother to child during birth, as well as through contact with blood or other body fluids, including sex with an infected partner, sharing or reusing syringes and needles. It represents a much more serious threat since it can lead to a chronic condition and may even progress into cirrhosis or liver cancer. Every year, more than 1 million people worldwide are subjected to death due to hepatitis B virus. It was thus a global health concern which had to be identified as soon as possible. And finally, in 1965, Dr. Baruch Blumberg discovered hepatitis B virus, for which he was awarded with Nobel Prize in 1976, as it was a significant discovery that saved millions of lives with the advent of diagnostic tests and vaccines. The non-A, non-B hepatitis virus. Even though Blumberg had discovered hepatitis B virus, there was still an unknown causative agent which caused chronic hepatitis. This pointed out a chance of some other etiology too, and there started the onerous journey that led to the discovery of hepatitis C virus. Harvey J. who was studying the occurrence of hepatitis in patients who had received blood transfusions, noticed that there were a lot of cases caused by an unknown infectious agent. As the vaccine and diagnostic tests were available for both hepatitis A and B viruses, they ruled out the possibility too. In a series of experiments done on various animals and models, Alter and his colleagues showed that blood from these chronic hepatitis patients could transmit the disease only to chimpanzees. Their subsequent studies also demonstrated that the unknown infectious agent had the characteristics of a virus. Alters, the studies ended by defining new, distinct form of chronic viral hepatitis that came to be known as non-A, non-B hepatitis, to isolate those DNA fragments derived from virus that codes for viral proteins responsible for causing the disease. They used the serum, that is, blood without clotting factors, of the patients infected with these viruses on the assumption that it would contain antibodies against the viral proteins. Based on reaction of the antibody to the specific disease-causing substance found in that blood of chimpanzee, they isolated of the DNA fragment encoding viral proteins. Further work showed that this DNA fragment was derived from a novel RNA virus belonging to the flavivirus family, and it was named Hepatitis C virus. The presence of antibodies in chronic hepatitis patients strongly implicated this virus as the missing agent. Potency of Hepatitis C virus. The discovery of Hepatitis C virus was decisive, but one essential piece of this puzzle was missing. Well, could the virus alone cause hepatitis? Okay, Charles M. Rice decided to solve this puzzle. He, along with other groups working with RNA viruses, took several samples of virus isolates and started studying the virus gene sequences in detail. One of the Rice's most important contributions was his recognition that the published viral sequence was incomplete. He noted a region in the end of hepatitis virus RNA that they suspected could be important for virus multiplication, which in turn leads to the disease. The breakthrough made it possible to engineer a version of hepatitis C virus capable of infecting animals and causing hepatitis to them. When this RNA was injected into the liver of chimpanzees, virus was detected in the blood and pathological changes resembling those seen in humans with the chronic diseases were observed. This work provided the final evidence that hepatitis C virus alone could cause the unexplained cases of transfusion-mediated hepatitis. Rice and others went on identifying mechanisms that the virus used to infect cells, multiply, and cause disease, and all potential targets of antiviral drugs. Rice then developed a system to screen drugs that block key steps in the viral life cycle, eventually leading to the development of curative drugs for hepatitis C virus infection. Significance of the discovery. According to the World Health Organization, about 71 million people in the world have chronic infection with the hepatitis C virus, which is also a major cause of liver cancer. In India alone, 40 million people are chronically infected with the hepatitis B virus and 6 to 2 million with the hepatitis C virus. Thanks to their discovery, highly sensitive blood tests for the virus are now available, and these have essentially eliminated post-transfusion hepatitis in many parts of the world, greatly improving global health. Their discovery also allowed the rapid development of antiviral drugs directed at hepatitis C. For the first time in history, the disease can now be cured, raising hopes of eradicating hepatitis C virus from the world population. Prevention, the first step to eradication. To achieve the goal of eradicating the disease from the world, international efforts facilitating blood testing and making antiviral drugs available across the globe will be required. Along with the spread of awareness about the cause of the disease, and preventive measures have to be taken. Being a blood-borne viral infection, the main ways of prevention include: avoid reusing or sharing of syringes. Make use of disposable syringes. Avoid sharing personal care items that might have blood on them, example razors, toothbrushes, nail clippers. If you are a healthcare or public safety worker, follow universal blood or body fluid precautions and safely handle needles and other sharps. Consider the risk If you are thinking about tattooing, body piercing, or acupuncture, make sure they are properly sterilized. Avoid multiple sexual partners and take necessary safety precautions to prevent sexually transmitted diseases, including that of hepatitis C. Along with adoption of these preventive measures, let us also hope for more faster research projects in the arena aiming towards complete eradication of the disease.